These are the lecture notes for Chapter 9 on the cell cycle and cellular reproduction, which is primarily going to be covering mitosis. So the sections are the cell cycle, and that's going to be the events from the time a cell is born. Basically, a cell is created until that cell divides or dies. And then the eukaryotic chromosome, we will look at, for example, humans have 46 chromosomes and our cells are eukaryotic. So different organisms that are eukaryotic are going to have different numbers of chromosomes, and we'll talk about that. Mitosis is a division of the chromosomes, the separation of the chromosomes into the two daughter cells that are produced when a cell divides. Cytokinesis is the actual division of a parent cell into two daughter cells. So mitosis is the division of the nucleus, and cytokinesis is the division of the cytoplasm and the plasma membrane. Then we're going to talk about the relationship between mitosis and cancer. And then just a short, we probably won't even cover section 9.5 besides just for me to tell you that when prokaryotic cells divide, it's called binary fission. Okay, so the cell cycle is an orderly set of stages from the first division of a eukaryotic cell to the time the resulting daughter cells divide. So the cell that's going to divide is called the parent cell, and then the cells that are produced when that parent cell divides are called daughter cells. So make sure you have this definition. The cell cycle is the stages from the first division of a eukaryotic cell to the time the resulting daughter cells divide. And there are two major stages of the cell cycle, and they are interphase and the mitotic or M stage, which includes mitosis and cytokinesis. This is a diagram or a graph, a pie graph, that shows you the different stages involved in the cell cycle and basically the average amount of time that the cell spends in each of those stages. So you can notice here that in purple, the majority of the cell cycle, the cell spends that in interphase. And interphase is going to include G1, S, and G2 stages. G1 is the growth stage that includes the organelles inside of the parent cell are going to copy themselves. So if there's one mitochondria, that mitochondria will make a copy so that the daughter cells can each have, the same number of mitochondria as the parent cell. So the first G1 stage is going to be where the organelles replicate. The S stage is going to be where DNA replicates. So the chromosomes, in other words, if there's 46 chromosomes in the parent cell, there will be 46 chromosomes in each of the daughter cells. So the DNA has to replicate or copy itself. The chromosomes which contain the DNA are going to have to replicate. And then the second growth stage is G2, and basically the spindle fibers are produced in G2. You notice in the M stage that you have mitosis, which consists of prophase, prometaphase, metaphase, anaphase, and telophase. And then after mitosis, you have cytokinesis, which is division of the actual cytoplasm. And what you're seeing on this page is checkpoints. So if wherever you see the stop sign symbols, if a cell does not meet the conditions at that checkpoint, it will not continue on. It will not continue to divide. And that is to protect cells from dividing when they are abnormal. Most of the cell cycle is spent in interphase, where the cell performs its usual functions. But you do need to know this, that nerve and muscle cells do not complete the cell cycle. They remain in the stage that we call G0 because they do not divide. nerve and muscle cells. Okay, interphase consists of the G1, S, and G2 stages. It takes up 90% of the cell cycle, so the cell spends about 90% of its time in interphase and only 10% in the M stage, mitosis and cytokinesis. In the G1 stage, the main thing I want you to know is that the cell increases its organelles. In the S stage, I want you to know that the DNA in the form of chromosomes is going to replicate. S stands for synthesis. So DNA synthesis or replication occurs. In the G2 stage, the cell is going to synthesize microtubules necessary for division, and we call these microtubules spindle microtubules or spindle fibers. So the cell is going to produce spindle fibers, and these spindle fibers are going to attach to the chromosomes and organize them during cell division, during mitosis. Now, what you're seeing here on slide number 7 is a chromosome that is not duplicated or replicated yet, and then you're seeing the replicated chromosome. So a replicated chromosome has the same DNA as the unreplicated chromosome. A replicated chromosome has two sister chromatids, and those sister chromatids are identical. They contain an identical copy of the DNA code, and they are attached at a place called the centromere. The centromere is like the attachment place, but those little we need to change this. I'm sorry. The centromere is the place where the chromatids are attached, but those structures that you see that are yellow or yellow-orange, those structures are called kinetochores, and those are going to be the structures that the spindle fibers attach to when we talk about spindle fibers attaching, and that's kinetochore. Sorry about that little mistake there. Okay, in the mitotic stage or the M stage, we have mitosis, which is the division of the nucleus, and then cytokinesis, which is division of the cytoplasm. Mitosis results in two genetically identical daughter cells. In other words, two daughter cells that are clones of the parent cell. This section is on controlling the cell cycle. We'll come back to it. But I do want you to know that there is a plan in place, or there's a process in place. It's called apoptosis. And apoptosis is programmed cell death. If a cell is unhealthy, if there's something wrong with it, if it has a mutation, if it's cancerous, if it's infected with a virus, just different reasons that the cell is not a healthy, normal cell, then it will typically not be able to make it through all of the stages of interphase or all of the stages of the mitosis stage. And apoptosis is caused by enzymes called caspases. Apoptosis is caused by caspases. Caspases are enzymes that will cause a cell to basically commit suicide. And it's committing suicide because it's unhealthy, or it's potentially unhealthy. So cell division, including mitosis, leads to a larger or greater number of cells. It increases the number of cells in that organism. Apoptosis decreases the number of cells. So this is just an image that shows you how apoptosis works. You have normal cells that blow up, that hypertrophy, basically, which means that they get larger, and then those normal cells then fragment. That's what happens in apoptosis. Okay, so we're going to skip now to section 9.2, which is on the eukaryotic chromosome. The eukaryotic chromosome is DNA, basically a single DNA molecule, which is shaped like a double helix, and the DNA is wrapped around histone proteins. So the DNA and the histone proteins are called chromatin. Now, during interphase, what you have in the nucleus is chromatin, but when the cell gets ready to divide, the chromatin will condense and it will form into a chromosomes. It will condense into chromosomes, and a chromosome looks like this. Chromatin is very spread out. So chromatin is going to be DNA wound around these purple histone proteins. They're purple in the illustration. But it's very, very spread out, very diffuse. And then that chromatin packs itself together. And when it packs tightly, it goes from being called euchromatin or true chromatin, which is not packed very tightly, into tightly packed heterochromatin. Now, euchromatin is able to do what DNA does, which is make proteins, but heterochromatin is packed too tightly to do that. So heterochromatin is the last stage before the chromatin becomes a chromosome. And chromosomes are visible under the light microscope. We can see them when we do lab. So heterochromatin is a very tightly packed form of chromatin, and it is inactive. Genes, what the purpose of a gene is to make a particular protein. So the genes are hardly ever transcribed into proteins. Heterochromatin does not. The genes are inactive. They don't do what they're designed to do. All right, now we're going to move on to section 9.3 on mitosis and cytokinesis. It is important that you know that mitosis is nuclear division, is the division of the nucleus. Cytokinesis is division of the cytoplasm. During mitosis, the sister chromatids are going to separate, and each daughter cell will get a copy, or get one sister chromatid from each pair. And before mitosis begins, let's just skip that part. This is just a table that shows you the chromosome number for different organisms. And I think it's just interesting to go through. You don't have to know these numbers, but I think it's interesting to see. Humans have 46 chromosomes. Humans or Homo sapiens have 46 chromosomes. And then the one that we're most closely related to, according to chromosome number, is a potato, which is Solanum tuberosum. So a potato has 48, but that does not mean we're more closely related to a potato than we are to a fruit fly. A fruit fly has only 8 chromosomes. A goldfish has 94. A fern, the southern adder's tongue fern, has 1,320 chromosomes. So hopefully you can see that chromosome number has nothing to do with how closely related the organisms are. You can have a large number of chromosomes, but still not have as much DNA, because each chromosome can be different sizes. So just because you have 1,320 chromosomes doesn't mean that they are large chromosomes. They can be very small chromosomes, and then you can have 8 chromosomes that are very large. So anyway, chromosome number really doesn't tell us that much, except that each species has a chromosome number for that species. Okay, and the chromosome number is diploid or 2N when there are 2 sets, one from the mother and one from the father. Humans have 23 pairs of chromosomes, so the diploid number is 23 pairs or 46. The 2N number for humans is 46. We have 46 total chromosomes. 23 come from our mom and 23 from our dad. Now, the haploid number of chromosomes is the number found in the egg or the sperm, and that haploid number for humans is 23.